My name is Dr. Eric Hilton. I'm a professor here at the Virginia Institute of Marine Science in the Fisheries Science Department. I'm also the curator of the Nunnally Ichthyology Collection, a unique resource that we have here at VIMS. What I want to talk about today is a little bit about both how we prepare specimens for specific research projects, but also then how we use them and why we use them. Most of the collection are stored in alcohol. So formalin-fixed specimens that are preserved and then await further study. This is a sturgeon from the Danube River in Europe, the stellate sturgeon. And this is one of the species that I've been studying the last few years in conjunction with a broad-based study of sturgeon anatomy and evolution. So we went there, we prepared the skeleton such that we could bring it back and feed it to our beetle colony. So this is one of the ways that we prepare and study specimens. We use dry skeletons, and you can see all of the little bumps and hooks and spines on this skull. Those are potentially informative for understanding the relationship between this species and other species around the world. When we prepare our specimens to be skeletons, we typically are looking at larger animals, and we use beetles to prepare them. So we dissect them, we dry out the dissection, and then the beetles, here you can see them on this knife fish that I collected in Florida, they go and they eat through all of the tissue that is left, preserving or leaving just a skeleton. And that allows us to study and lay side by side different species and compare them to see how they are similar and how they are different. These dry skeletons are very informative and can be very important for studying large animals. But one of the other interesting aspects of comparative anatomy of fishes is that we can understand and study their entire life. We can study from their earliest stages all the way up to large individuals. On this smaller specimen, this is a cleared and stained specimen, and those specimens start their lives, if you will, as a preserved specimen, and then we soak them through a series of chemicals and dyes to stain the cartilages blue and stain the bones red, and then we can make all of the soft tissue, render it clear, such that we can see through and see the skeleton as a whole structure. And then we can compare this 6-inch long specimen to this 3-foot long specimen, and we can get a sense of how that has changed across their lives. And that can be very informative. We're able to see and study each individual bone as it develops. And that has led to some really interesting discoveries that had us rethink how the skeleton of toadfishes is constructed. By using these cleared and stained specimens, we're able to understand how that change has happened, when it happens, and what that might mean for the evolutionary history of that fish. One other example is of how we use these cleared and stained specimens. This is a small individual that has been cleared and stained of that stellate sturgeon. One of the unique things about sturgeons is that when they're larvae, they have teeth that then fall out in their early life history. This individual has just lost all of its teeth, but you can see where those teeth inserted into their jaws. And what that tells us is at this size, this little sturgeon was still eating animals, little tiny zooplankton animals that they found in the rivers. And that is an interesting piece of natural history, piece of information about their biology that can tell us something about how to conserve them. That's just another example of how we use these specimens for understanding the biology of fishes generally.